Good morning, children. So already you know that we have started with a new chapter, that is diversity of living organism, or can tell it as classification of living organism. Good morning, children. Rachita, good morning. Shravani, good morning. So good morning, everyone. Once again. So after a long gap, so science class is taken. So in the last previous classes, we have learned about classification. We have learned about classification, then how it is classified, on what basis it is classified, then hierarchy of classification we have learned. Then in this class we are going to study about the first kingdom, that is Monera kingdom. Before that, already you written the exam test you have written yesterday. I hope you have done very well. Some of the answers I have seen. The source whatever you used is different. Okay, leave it. That we'll see it later. Okay. Yeah, Kingdom Monera. So all the organisms which come under Kingdom Monera are prokaryotes. Prokaryotes means they are organisms are made up of prokaryotic cell. Prokaryotic cell. All are prokaryotes. That is the main. That's the prokaryote is the main characteristics of the kingdom Monera. Kingdom Monera. All are prokaryotes. So examples, if we give examples, all bacteria comes under this. All bacteria, because they are prokaryotic, means the nucleus is not covered actually. The genetic material inside the cell is open type, means it is not covered, protected. So bacteria, blue-green algae. This is an example. Then take, they are present. They habitat in all type of environment. All type of environment means they are some bacteria they are present in living some other body of living organism, like in our body also. In our body also there are some bacteria which grow in our body, but they do not harm us, like that. So there are bacteria which are called opportunistic bacteria. If your immunity comes down, they may harm you. On our skin also there is a bacteria called Staphylococcus. In our inside the body there are lactobacillus. In intestine there is a lactobacillus. In body, E. coli is there. Like that, many of the bacteria reside in our body, in other animal body also. If you take ruminants are the animals which will do the mastication or ruminating process. Agiyoke ಅಂತ ಹೇಳ್ತಾರಲ್ಲ, ಅಗಿಯೋದು ಅಂತ ಹೇಳ್ತಾರೆ. ದನಕರುಗಳು ನೋಡಿದೀರಾ, ಮೇವು ತಿಂದಾದ್ಮೇಲೆ ಬಾಯಿನ ಅಗಿತಾ ಇರ್ತದೆ. So those are called as ruminants animal. These ruminants will have some bacteria which will help in the digestion. Not only that, in our body also there are bacteria which help in digestion. Like that, if you take, they are present in water also, they are present in air also, they are present in soil also, they are present in the body of other organism also, and They are also present in, and there are some bacteria which can survive in a cold environment, also hot temperate environment also. Means they are present everywhere. They are present everywhere because they are capable of surviving in all type of environment, everywhere. All the possible habitats ಅಂತ ಹೇಳ್ಬೋದು. Marine, salt water ಇದೆ, cold water, whatever it is, cold water, hot water. So some bacteria, thermophilic bacteria ಅಂತ ಹೇಳ್ತೀವಿ. Even in the cow dung there is a bacteria. Even waste ಅಂತ ಹೇಳ್ತೀವಿ ನಾವು. ಅಲ್ಲೂ ಕೂಡ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇದೆ, ಮಿಥುನೋಜೆನೆಸಿಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಅಂತ ಹೇಳ್ತೀವಿ ನಾವು. ಸೊ ಕೌಡಂಗ್ ಇಂದ ಮೀಥೇನ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತಾರೆ. ಆ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಮಿಥುನೋಜೆನೆಸಿಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ. So in the cow dung there is a bacteria called as methanogenesis bacteria which produces methane gas. Like that, everywhere there is a bacteria. That's why they are living all type of habitat. All type of habitat. Then nutrition habitat. They are autotrophic. Some are autotrophic. Some are autotrophic in nature. Autotrophic means they do chemosynthesis. Chemosynthetic bacteria. They produce the food. They produce the food in the absence of oxygen also, the presence of oxygen also. So they do the aerobic respiration also, anaerobic respiration also, and they do the. So they prepare their own food also. Some bacteria prepare their own food by the help of chemosynthesis. Chemosynthesis means preparation of the food by the help of some chemicals. Chemosynthesis. Some are, I told you, this blue green algae. Blue green algae actually it is a photosynthetic. Photosynthetic it is. See, it is also there. And there are heterotrophs also. Heterotrophs. Heterotrophs, bacteria are more. Heterotrophs include saprophytes. Saprophytes are there. Parasites are there. And even one more is there. There is symbiotic bacteria is there. So there are different type of mode of nutrition. This is nothing but mode of nutrition. Particular mode of nutrition, preparing their own food by using some chemicals or sunlight. So even photosynthesis also happens. I told you, some bacteria. So heterotrophs. There are many saprophytes. The bacteria which grow in the decaying materials. So bacteria which grow in the other animals completely for food as well as shelter. There are symbiotic bacteria. I told you E. coli. E. coli and the bacteria in the bottle. E. coli. That is symbiotic bacteria. Parasite. Parasitic bacteria. There are many parasitic bacteria. Staphylococcus. Don't try it. So there are many bacteria which are given such examples. Then, if you see types, so bacteria are divided on the basis of shape. So, this classification it is on the basis. So bacteria are round shaped, it's called as coccus. If they are round shaped, they are called as bacillus. If they are spiral shaped, then called as spirale. They are comma shaped, we call it as vibrio. There are like coccus, bacillus. Lactobacillus. That give an example. So spiral bacteria are there. Commercial bacteria called Vibrio cholera. Cholera disease is caused by Vibrio commercial bacteria. So spiral bacteria are there. All these types of bacteria are there, and So these are the names are given on the basis of this shape. If it is round, compass. If it is rod shaped, bacillus. If it is spiral shaped, spiral egg. If it is comma shaped, vibrio. Like that, we have a different type of bacteria depending on the shape. And she also divided on the basis of gram staining. On the basis of stain means to see the microorganisms in microscope, we will actually stain it. Stain means we will use some chemicals which are colored chemicals to identify the bacteria. So gram staining ಅಂತ ಹೇಳ್ತೀವಿ ಅದಕ್ಕೆ. On the basis of gram staining, we call positive, gram negative, gram positive. So gram negative, gram positive. You know, we will not go through this. What is this in depth? You understand that on the basis of gram negative, positive bacteria, negative bacteria are there. So you will learn in higher classes about this. So this is about the some characteristics of the kingdom Monera. If you see the structure of bacteria, which is one of the organisms which come under Monera kingdom. So how they are taken as, we take bacteria, bacillus bacteria. So they will have genetic materials. Genetic, which is made up of DNA. Understood? They have flagella. They move by help of flagella. The structure called flagella. They have vacuoles. They have cell wall. Cell wall is that, and they are covered with a thick capsule. Capsule is there. Protective layer is there. Cell wall is there. Cell membrane is there. So all this like vacuoles are there. Vacuoles are there. So all these are all the cells contains, no? Right. But they do not have all the organelles. Like mitochondria is not there. Mitochondria will be not there. Means say prokaryote, eukaryote. Prokaryote will not have this. All the organelles will be not there. Some organelles will be there. Some are not there. Okay. That's it. So they prepare. So they do the. See, mitochondria will be absent over here. Mitochondria, what it does, it produces the energy. But in this, mitochondria will be absent in prokaryotic cell, and it will be done by the production of energy is done by the plasma membrane itself. Understood? So, like that, ribosomes are there. Ribosome protein synthesis. Ribosomes are, you know it. They are called as protein factories. Ribosomes are called as protein factories of the cell. Means protein required for the cell is produced by the ribosomes. Ribosomes are there. Some organelles are not there. Some organelles are there. This is the structure of the bacteria. If you take the simple bacteria, and moreover, the cell wall actually made up of peptidoglycan. That's a very important point. Cell wall is made up of peptidoglycan. Abhirav, whatever video is possible. Maybe your network problem at. For others, it is not happening. Aditya and Abhiram, most probably network problem in your area. It will be sorted out. Wait for some time. So this is about the structure of bacteria if we take. I hope you got the point. This is about the structure of bacteria. Usually, this is about the bacteria and bacteria means kingdom Monera. You concentrate on Monera. These are the characteristics of the Monera. The organism which fall under or come under the Monera kingdom will have these kind of characteristics. If you take. Understood? So I told you about the nutrition also. I told you about the habitat also. We talked about. We discussed about the structure also. So we say now how they reproduce. How they reproduce. They reproduce by like they reproduce by sexual methods. Sexual method both sexual. Sorry. They reproduce by sexual method as well as asexual method. So both are followed. So budding is one of the sexual reproduction, asexual reproduction. Then we have binary fission. Fission is one of our sexual reproduction. Like that, there are many types of reproduction. So like that, they reproduce by methods called budding. As well as asexual reproduction, like budding also takes place. So bacteria are the organism which usually reproduce by both the methods. Yeah, like that. So both are there. So endospore, there are many methods. You remember budding and fission. Like endospore, one more is the endospore method. Means see, when unfavorable condition comes, unfavorable condition means when the bacteria, so when the environment is not suitable for the bacteria to grow, then it's called unfavorable condition. Favorable condition means what? The bacteria can grow very easily. That condition is called a favorable condition. During the unfavorable condition, they produce a capsule. They produce a protective layer, and that protective layer with that bacteria is called an endospore. When favorable season comes, it will generate into new bacteria. So like that. So asexual reproduction. So most probably asexual reproduction. Sexual reproduction happens like they just transfer the genetic material. They transfer conjugation ಅಂತ ಹೇಳ್ತೀವಿ ನಾವು ಅದು. Conjugation by the method of conjugation. Just transfer. That method is also done. Usually asexual method is the most happening in a bacteria. Is it clear? This is about the bacteria. And if you take, see, importance. So bacteria are important for us. What is the importance of bacteria or Monera? So they help, say, they help. So they are harmful as well as useful. Harmful as well as useful. They cause many diseases in plants and animals. In plant and animals. Example. Many of the examples you can give here. Many of the examples you can give. Typhoid you can give. Pneumonia you can give. So many of the examples you can give. Cholera you can give. Example cholera. It's caused by Vibrio cholera bacterium. Cholera bacteria. Pneumonia. Pneumonia is caused by mycoplasma. So, mycoplasma. So there are many bacteria which causes the disease also. There are many bacteria which are helpful. So they are helpful also. Yeah, helpful also. They help in the nitrogen fixation. They help in nitrogen fixation. Means so atmospheric nitrogen. The nitrogen which is present in the atmosphere cannot be used by organisms which are terrestrial organisms. Like we cannot use directly the nitrogen. Our plants cannot use the nitrogen for the preparation of protein. We need nitrogen for the preparation of protein, but we cannot take up the nitrogen directly from the atmosphere. That has to be converted into nitrates. That conversion is done by bacteria called nitrogen fixation bacteria, like rhizobium. Rhizobium. So lactobacillus actually help in setting up curd. So milk is converted to curd by the use of lactobacillus. They are used in beverages to prepare beverages. So they are used in leather industry. Leather industry. They help. So they are used in preparation of antibiotics. Preparation of antibiotics. Antibiotics are also prepared from this, which are helpful in to kill the microorganisms which causes the disease. So disease causing microorganisms are called as pathogens. Pathogens cause many diseases. So like that, if you take, there are many uses of bacteria. If you take, they help in the decomposition of waste. Organic waste in the sense organic waste, plant waste and animal waste, decomposition. So without this bacteria, decomposition will never happen. So plants need manures. Manures, how they are produced? Manures are produced by plant waste and animal waste that are decomposed. Who decomposes that? Bacteria decomposes, fungus decomposes. So that's why they are helpful also, they are harmful also. Understood? So like that, if you take, there are many number of examples you can give. So tuberculosis is caused Mycoplasma tuberculosis. Like that, many of the diseases are caused by plants and animals, and they are very useful also. If you take. So this is about the kingdom. Monera, go through your textbook and read it. If you have any doubt, you can ask. So I will end the meeting. One more meeting link will be sent. Join with that meeting link. Or join with the same link. One more meeting. Thank you.